ion channels, right? And there would be some sort of stimulus coming in, effecting some sort of change, right? Whether it might be ligand gated, it might be voltage gated, it might be mechanically gated, right? Right? We know those are our choices. So something's going to come in, effect the change, and perhaps open the channels, and something will happen in the neuron, like a graded potential. We know that, right? That was our example. Right? So we know that. All right. Our other setup could be that here's our neuron, right? And we've got our ion channels just like we've already set up, right? But we might have a little separate cell first, right? Sort of in between the stimulus, whatever that is, and this neuron. It's still in the nervous system, right? We're still going to classify it as a neuron, but this is going to be a very, very specialized neuron. Let's call it a receptor cell. So keep in mind as we use the word receptor, right? Now in this part of the nervous system, so far we've thought of receptors as, some of these channels, right? Because they could receive, let's say, a neurotransmitter, right? But now we are going to have perhaps a whole entire cell that is highly specialized, and we'll call that a receptor cell. But keep in mind that a lot of times as you're reading, or maybe as I'm talking, or maybe somebody else is talking, they might say receptor, and they might be referring to this whole entire cell, right? So we want to be sure that we're aware, right? That sometimes people just say receptor, and maybe technically they're talking about this whole entire cell, that its job is to receive that information, create some sort of graded potential, and then release some neurotransmitter onto this big neuron, right, that's going to send. Good? So we have it's like a middleman, right? The receptor cell is going to be like a middleman for us. Good? But just don't get confused if, if somebody, including myself, just says receptor, right? Sometimes we're talking about this whole entire cell. And we'll see examples as we go through all the different senses. All right. Let's see. Here we've got some classes of receptors. There's our list. Mechanoreceptors would respond to maybe shape changes, right? Pressure, touch, something like that. Stretching. Thermoreceptors, temperature. Photo, light, right? Chemo, any sort of ligand, right? A chemical. And nociceptors are for pain. So maybe noci might be a new word for you, but that would be a pain receptor. Each receptor is going to be extremely, extremely sensitive to what it is built to receive, right? So let's say a light receptor or photoreceptor is very, very, very sensitive to light, right? And we'll talk all about light, I think that's tomorrow, actually, when we get into the details of that. But, so you'll need only a little bit of light to get that receptor to respond. Good so far? Having said that, each receptor can respond to other things, but if it's going to respond to some other thing, it's going to take kind of a lot. So as an example, since we're talking about light, right? If our eyes are open and light is present and it's entering into our eyes, details tomorrow, that will be received, right, by our photoreceptors on the retina. And even just the tiniest, tiniest little bit of light, right, in the darkest room, that will still register, right, to those photoreceptors, and we'll get a little bit of a response, right, if it's a little bit. But we will get a response to that tiny little bit of light. Let's say, you know how, actually whether your eyes are open or closed, I was going to say, when you close your eyes, but it's more obvious when you close your eyes. But it'll still happen if your eyes are open. But if you close your eyes and you press on your eyeball, like you can do that now. If you close an eye gently, like press on your eyeball. I think it works better when you close both eyes because then you're not seeing all this other stuff. But you see how you do sort of have a sensation of light? Like it might not necessarily be like, oh, like I can everybody here and what everybody's wearing and all that stuff, right? But you do get like maybe sort of these flares of greenish, yellowish, bluish, right? Blobs when you press. Make sense? So that's an example of a different stimulus. In this case, it's a pressure, right? A pressure stimulus activating those photoreceptors, right? It kind of takes a lot. Like, I don't want you to press really hard, but you do kind of have to press kind of hard to get those blobs of light. But that's an example of those receptors responding to something that's not light, right? Something that they're not built for. But do you notice that the sensation or what we receive, right, because of that different stimulus is still something that has to do with image, right? It might not be, like I said, it might not be seeing you guys and whatever, all the different colors, right? And clothes and all that stuff, right? It's just sort of blobs. But the blobs of light are still registering to me as blobs of light. Make sense? All right, so that's the thing about these special receptors. Good? Let's see. Here we've got a word. My stick. Sensory transduction. Oops. Sensory transduction. So this is going to be all these various processes that different receptors are going to use that we will go through one at a time for each sense, right? The process where that receptor takes that stimulus in whatever form it arrives and turns it into that electrical response that we learned about, right? And it's usually going to be that graded potential, right? Graded potential. And now we're going to use, instead of the word graded potential, receptor potential. So keep in mind also those two words will be interchangeable. Like sometimes we can still say graded potential. That's fine. But if we use the word receptor potential, we know that that is a graded potential that we already have learned about. Good? And we also know that a graded potential could be this direction or this direction, right? Depolarization or hyperpolarization. Good? Either way, it'll still be a receptor potential. All right, very good. And we already know, right? That's obvious. Ion channels. All right, so let's see. Oh, I hate when the top is cut off. It's like, where's the title? I like the title. Anyway, this is kind of a summary slide that takes information that we already know, right? And now we're going to sort of add and build and look at it in a little bit of a different way. So we've got a stimulus coming in, and we've got our ion channels, right, our receptors on this membrane of this afferent. Remember, afferent is a neuron coming in, neuron. And then we've got our axon. They're telling us this one is myelinated. Fine. This just means it's really long, right? And then we get to the other end with our neurotransmitter. Good? All right, so that's our setup. And they're also showing us that they are measuring voltage at these different locations. So we've got here axon hillock. And what are we going to do at axon hillock? Sum, right? We're going to sum. All right. And here, we've got a node of Bouvier. What are we going to do there? Action potential, right? That's our only choice. So we're going to measure there. And then, okay, we go further on down the axon. Kind of the same thing, right? Action potential. But let's see, as we approach the end, right? Kind of thing. All right. So here we've got on this first, so we've got all these lines, right? Sort of little mini graphs kind of thing. So we've got these three different intensities of stimuli. It's the same stimulus, right? But three different intensities. So kind of weak, kind of in the middle, kind of big, right? Like baby bear, mama bear, papa bear. Good? And we see stuff that this is what we already know, right? We could, especially after summing, end up with some sort of voltage, right? Some sort of size of gradient potential, right? As our sum. That's what this is representing. So this little stimulus, we ended up with that, this medium size, right? And then the big. Totally makes sense. We already know this stuff. All right, from here, that was at our axon hillock. And if, remember, we reached threshold, the cell was going to create an action potential, and once you create that action potential, it's going to get sent where going, right? So let's see what we end up doing as far as action potentials are concerned, depending on the sum, right? So it looks like here we had a small sum, and well, it looks like we didn't reach threshold with that one. So we know, stuff we know. Looks like here we reached threshold, and sort of at this sum, it turns out that, oh, we're not going to create just one action potential. Maybe this is actually enough, right, of a graded potential that, okay, they're giving us 6 action potentials. Like it was strong enough to create 6 action potentials, not even just one, right? But 6. That's pretty good. All right, let's see. And then now there's a really big one, right? Oh my goodness, look at that. We're creating a whole bunch. I can't even count that, right? A whole bunch of action potentials. So we see we're going to add this new little bit of information to what we already know. It turns out, depending on the sum, right, beyond threshold, like so far we already know you don't hit threshold, no action potential. You hit threshold, action potential. But we can add to that and say, well, if the sum is big enough above threshold, we could create more than one action potential. If it's really, really enough beyond threshold, even more, right? So the number of action potentials that we're going to generate, right, is dependent on how far above threshold did that sum end up being. Good? All right, that should totally make sense, I hope. All right, very good. And then we travel, right, down the axon, and they just pick some node, and let's see what has happened. It turns out this we should already know, but this is just to reinforce, to be sure. We should have the same, right? Because of all that myelin, nothing should have dissipated, right? So we should have the same, and we do. Number of action potentials, right? So this will stay constant, this will stay constant. When you move further down.
May have nothing to do with it, but like the refractory,
Yeah.
like I thought you're saying it generates 6 actions, but like after the first one's complete, like after the hyperpolarization, it starts another one. So they're.
I think I see where you're going. These are going to happen pretty much all at the same time. Because remember, this is a 2-dimensional drawing, but of course we're 3-dimensional, so that axon hillock is really this, like a cone shape. So we've got all these patches of membrane that we can use, right? So let's say we've got this cone, we can easily create 6 action potentials anywhere around this cone at the same time, right? Right? And then they can all travel together. Does that make sense? And I think that will sort that out. It's not like create one and then the second and then the third, right? And then you've got your 6. Let's do them all at once. Good. Sort of bound. Good. Very good. All right. And we know this at the bottom, no action potential, and nothing gets released, right? No neurotransmitter gets released. Here we see we've got our 6 arriving, and we'll release some neurotransmitter. Here, if we have many, many more, right? I think this might be double. Wow, we're going to release more neurotransmitter. Make sense? All right. So that's another little bit of new stuff. Whoops. All right. This is just to reinforce that particular picture showed us one axon. If we're in the situation where we've got our specialized receptor cell, right, kind of in the middle there, that's all right. We're just going to see, like, okay, something special is going to happen at that cell, and then all that stuff is going to happen at the long node. I don't know. I think that slide kind of states the opposite. Yes. All right. So anyway, let's now look at a process. It got cut off at the top, but it's adaptation. I should write that so you guys can see. Can I get my stuff together? So adaptation is a new vocabulary word. Adaptation example of this slide. You're not seeing it. And we are going to have two different types of receptors to, you know how, like, let's say when you first sit down in a chair, right, and you have that sensation on your skin, right, of the pressure of sitting in the chair. But now that you guys have been sitting for a while, right, and especially we do other stuff, right, where you're kind of looking and listening and all this stuff, right, hopefully not falling asleep or anything, but all this other stuff. And then, like, you're not so terribly aware of that sensation, like what it felt like on your skin when you first sat down, right? That sensation kind of, I don't know, it almost goes away, right? And then when you get up, right, when it's break time, we'll all stand up. When you get up, then, oh, there is a new sensation, right, on your skin when all that pressure from sitting is released, right? So that's a little bit of what we're talking about here, especially that particular example is in this top situation that we're looking at here. So we're going to have a receptor type in certain locations called a rapidly adapting receptor. And this example of sitting down, and then you sit for a long time, and then you get up would be kind of a classic example of a location where we have this type of receptor, this rapidly adapting type of receptor. So what happens is when you first sit down, right? When you first sit down, this would be our summed graded potentials, right? At those locations in the skin. All right, there's our sum, sitting down. And that will generate a whole bunch of action potentials. And you can even, if you really look at that detail, you'll see like, even among this group, towards the beginning, because this is time, towards the beginning, wow, we're really clustered. We have so many action potentials. And then they get a little bit further spaced out, right, in time. Good. But we do get to a point in time, and it's pretty soon, where, whoops, none, right? No action potentials, and that's because, no, we're not even reaching threshold, right, with those graded potentials. So, the receptors are quiet, right? We're kind of allowing that information to just like, it's so not important. Like once you're sitting, I mean, if you think about it in the big picture, like once you're sitting, it's not really that important in the big picture to keep telling your brain, you know, that skin on your butt is still being pressed on, right? Because once you're sitting, you're probably going to do other stuff, right? In the big picture. So why even bother sending all that information to the brain? And it's a distraction. Make sense? All right. So that's kind of the reason why we have these receptors in certain locations. Because, all right, once you're sitting down, move on. So, all right. So, okay, sitting, and now we're going to get up, right? But that's a change, getting up. That's a different sensation. Different stuff is happening at that skin. So, turns out those same receptors are going to generate some more graded potentials, because that's a change from what we just had. So we've got graded potential. It's enough to reach threshold, and that's enough over threshold to generate some action potentials. All right. This getting up thing is not as big, so we don't have as many, right? Action potentials. That should all make sense, but at least it's something, right? But that's important for us to know. If we're completely changing position, we should tell that to our brain, right? This is very sensible. We're going to call this stuff the off response. Off response. Even though, so don't get confused, even though it seems like, oh wait, but we're turning back on, it seems like on, off, on. Okay, I know that would be more intuitive, but no, that's not how they did it. Whoever decided to do the names decided to say, well, this is the off response. And maybe we could remember it as like actually sitting is on, and then when you're not sitting anymore, like the sitting is off. I suppose the opposite, slowly adapting receptors. And our big example would be receptors in the joints that are going to tell us where are our body parts in space, right? Is the joint flexed or extended or somewhere in between, right? And we're going to learn more about these. Where goes my shot? In the next section with muscle tissue. We're going to group those. So we'll learn about the details of these receptors next week, but we can talk a little bit about them, right, with a little bit of knowledge for today. So anyway, our joint positions. That's probably pretty good, right, in general, to be giving that information to our brains kind of all the time, right? Just so we know, right, and just in case there's some sort of change, right, like you fall over or something, right, something like that. Anything. That's always good information to know. So, we're going to put these different types of receptors in that location, and those receptors are, basically they're going to keep creating graded potentials as long as that joint is doing something, right? As long as we've got some sort of position in the joint, let's just keep making graded potentials. So if we're going to keep creating graded potentials that are high enough above threshold to create action potentials, great. All right, we're going to do that. Good. All right. For any given position, the beginning of the position, all right, so we'll have a little bit of a higher sum, and we'll have sort of a concentration of action potentials, and we might, as we sort of hold that position, right? Okay, we might sort of calm down a little bit. So we'll space out those action potentials a little bit, right? Calm down a little, but we're going to just keep going during that whole time, right? Unless until that position like completely changes, and then we'll do something new, completely new. All right. So here we see they're just showing us this is the stimulus, right, for this bottom thing. We're doing something, and then we're off. All right. So this is our little look at this other type, slowly adapting receptors, and we'll see these two types of receptors in different locations. All right. So it's just our intro to that. All right. So here we've got an overall general look at a sensory receptor. And we recognize skin. Right. Skin. Ooh. What is this part of skin? Anatomy review. Just in general, this superficial layer made of many layers. Epidermis, right? And that would make this deeper layer. Huh? Dermis, good, made of connective tissue, right? And we won't get into the details. You guys know that stuff already. Okay, very good. So we're looking at skin. We've got our sensory neuron here, and they've got our little symbol meaning, okay, this is going to be really long, and we're making our way towards central nervous system, right, in this bluish section here. Let's also review. So here we would be in spinal cord, right? Let's review. Aha, the structure of a sensory neuron. Now that we're looking at skin, remember we had this type of setup where we had that kind of floating balloon shape cell body, right? Remember that? Where was that located? Another anatomy review. Where did we cluster all of those cell bodies? Huh? Entering into the central nervous system, right? We're entering into the spinal cord, and we clustered all of these cell bodies for sensory. What was a ganglion? It was a something ganglion. Huh? Let me give you some clues. It was it's like towards the back side. Dorsal. Okay, dorsal what? Dorsal something ganglion. Dorsal root. Woo! Oh my god. So dorsal root, right? Oh man. Anatomy review. Dorsal root ganglion, right? is where we cluster all of those cell bodies for these sensory cells. Okay, so we're going to put that physiology and that anatomy together. All right, very good, very good. And then we continue, right, actually into the cord itself, and we'll bring that information here and release some neurotransmitters onto the next neuron in the chain. All right, so far so good. Oh wait, before we leave the slide, let's bring our focus back out to here, and we've got all these terminals, right? These they're sort of like axon terminals. Sensory neurons are kind of unique there. But we've got these terminals. These are going to have our sensory receptors on them, and you see where they've made this sort of orangey section here? Let's give that a name, receptive field. So this receptive field would be the area that the terminals of this one neuron is responsible for, right? This neuron is responsible for this area because all of these terminals, well, can sort of reach that area, right? So we'll just say, okay, receptive field for this neuron. All right, not bad, not bad. We could, and we will, we're going to see in the next picture, we will, I don't know, put a whole bunch of these sensory neurons like all next to each other, right? We have a whole bunch here. We could cluster together a bunch of similar neurons that are sensitive to the same thing, let's say pressure, right? And or I guess, we could also put next to each other different types of neurons. Maybe one would be sensitive to pressure, and the one next to it would be sensitive to temperature, right? So we could do either one of those things, and we do. So we would have our receptive field. Let's say this one's for pressure. We could have our receptive field for pressure here. Let's say if right next to that on this side we put a neuron that's responsible for temperature, right, and specifically cold, like freezing temperature, right? We'll put that one here, and let's say that one had a whole bunch of course, terminals out here, just like this one. All right, maybe they're intermingled, all those terminals. So those receptive fields, you see how they would overlap, right? If we've got the terminals of the adjacent neuron, like kind of intermingled. All right, so we could have something for touch with its receptive field in this location. We could have something for freezing cold right next to it with some of those terminals intermingled, right? It would be kind of like, right, like this. So that these two receptive fields, see how they overlap, right? So this enables us to receive information from a cold ice cube that let's say you have I don't know, big boo-boo, right? And you take the ice cube and you press it onto a patch of skin. So you can feel the coldness and the pressure at the same time in that same location because receptive fields are going to overlap. Make sense? All right, let's see. Okay, also, if we look at any one particular neuron and they're giving us 4 terminals. In real life, there are going to be hundreds of terminals, but here they're giving us 4 just to make it simple to look at, and they're giving us this glass probe just to give us something that we're going to press onto the skin. And here we don't press, so there's nothing, right? No sensation. Here we press a little. Maybe that's just enough to get some graded potentials generated in just one, right, of those terminals. And you press a little more, right? You see that? And so this is all this is saying is that the greater the stimulus, right, the greater the sum of graded potentials, and then eventually you end up to what we already know, right? You can generate more action potentials. All right, so let's see. Here, let me double check one thing. I'm just waiting for an answer. Oh, I guess so. Okay, hang on. person's gonna take over for me. All right. So now this is something we're gonna work on actually in our lab. So we've got something in the lab we're gonna refer to as 2-point discrimination. 2-point discrimination. So what this is, here we're looking at touch in the skin, and in different areas of the skin, you know how most of us know this kind of intuitively. Some parts of the body are more sensitive, right, than other parts of the body, and there's a reason for that, which we talked a little bit about in anatomy. It depends on, in the big picture as humans, what is really important for our survival, right? In general, our hands and fingers, right, are really important for our survival. So we should have a lot of sensation, right, able to come in on our hands and fingers. Same thing for the face, right, and especially the mouth. Very important for us, for our survival, to be able to eat properly and to be able to communicate. Because as humans, we really communicate a lot, right, with our face and especially mouth. Good? So it's really, really important to have a lot of sensation coming in from those particular areas, as opposed to, like, I don't know, like back here, right? And on your shoulders or maybe your back or something. Kind of important, but not as important, right? So maybe the sensations coming in from your back, right, your shoulders, we could kind of lighten up, right? We don't have all that much stuff coming in from there, because we could survive, right, without too much stuff coming in. So we've got this set up. So how can we set that up, right, in an efficient way? So let's look at two things. Here we've got an example of a neuron with a very small receptive field. And then, turns out, kind of nearby, we have a different neuron, and this one has a much bigger receptive field. All right, so far so good. Now let's look at this picture, and we see here on the lips, we've got 4 neurons that can fit into this particular space, and those 4 neurons all have the small receptive fields. Good? And we can really pack in a lot of neurons if they all have those small receptive fields, right? You put more neurons in because they're just small, right? At least the receptive fields are smaller, so all those terminals don't take up so much space. So we really pack them in. As opposed to here, they're telling us this is on the back. All right. Here, we're going to use those ones with the bigger receptive field, and wow, you can't pack so many in because those receptive fields are taking up so much space, right? All right, so we compare the two situations, and we're taking this set of calipers, and you just, you can kind of make it whatever distance you want between the 2 points, and you just keep that constant. And this is how we'll do the test, is, you just kind of poke lightly, poke the person, and just measure. Does the person tell you, oh, I feel 2 points being poked, or I only feel 1 point being poked, even though you are poking those 2 points, right? So let's look at this situation first. If we have, and you see how this is the same distance and this is the same distance, right? So you keep, that's the important thing, you keep your instrument the same. And we're going to poke here on the back. And when we happen to poke here, we see that both points are poking within this one receptive field, right? And not at all. They're not even close to this receptive field. So these 2 points are going to, sure, they're going to create graded potentials right here and blah, 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 all the rest, right? But this is only 1 neuron. So only 1 neuron is eventually going to make it right to the spinal cord and then up to the brain, right, send signals up to the brain to tell us, oh, something's poking me. But if only 1 neuron is doing that, we're only getting 1 sensation, right? So it seems like it's only 1 point. It will register, even though it's 2, right? It will register to our consciousness, right, as 1, because 1 neuron generated those graded potentials and action potentials and sent that signal up to the brain. Good. Remember homunculus? I think I have a review coming up. Remember homunculus, that mapping, right, of that somatosensory strip? We'll do that. All right, let's see what else. Take the same measurement, right? And now we're going to touch here. We see the lips, where we've got a lot of neurons with the smaller receptive fields. And keeping this the same, when you touch, looks like one of the points generates some signals here in this red neuron, and the other point hits the one next to it because they're small, right? So if we're generating graded potentials and then action potentials in two separate neurons, we're sending two separate signals, right? Eventually up to the brain. Make sense? So if we're receiving in the brain, right, primary somatosensory cortex, if we're receiving two separate signals from two separate neurons happening right next to each other, fine. That's gonna be perceived as 2 points. Good? 2 points. So, that translates into being more sensitive, right, here on the lips, being able to say, right, let's say eyes are closed, right, and the person does the, right, that test. You could tell the person, oh yeah, I felt 2 points here, but then you take the same thing and somewhere else, I only feel 1 point. Good? All right, let's see. All right, kind of having to do with that to help us to localize where exactly was that point, right? It was 1 point, or where exactly were those points if there were 2 points, right? Doesn't matter. Where was it? Good. We're going to have a setup, some kind of a setup, and we'll call this setup lateral. Got cut off a little bit. Lateral inhibition. Lateral inhibition. So our setup is going to involve some interneurons. Now we're going to add interneurons, and we're going to use inhibiting interneurons. So the red tells us, for our book, the way they set it up, that these are inhibiting interneurons. So an inhibiting interneuron would, based on what we know, from what we just got tested on, probably creating, right, graded potentials that are the hyperpolarizations, right, the more negative, the inhibitory, IPSPs, right? We know all that stuff. Green in these types of diagrams are indicating to us the EPSPs, right? These graded potentials. Good. So we're going to take 3 neurons and we'll put them right next to each other, like physically in the skin, boom, 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 right next to each other. And let's have some branches, right? Some branches so that they can reach out to each other. And we'll put some interneurons, the inhibitory ones, kind of right in between, so that the inhibitory neurons can influence all of these branches, and then the neurons that are next to them. All right, so let's see. We'll look over here. So we've got, no, actually, let's start here. Let's start in the middle. Let's look at the neuron that's in the middle. And let's say we are going to poke, right? So I'm going to take this pen here, and I'm going to just poke the skin, right, on this particular neuron. So I'm going to poke here. And this neuron is going to, feel that, right? Just right, pressing. It's going to feel that and send some signals. And wow, maybe I pressed really hard, so we're going to generate a lot of action potentials. Okay, great. So we'll send that, and here we're coming to the end of the neuron, and we've got our branches. So we'll send that signal, all those action potentials. Let's go in all those directions. We'll just follow the branches. No big deal. All right, let's keep going straight. When we hit this interneuron, let's say now we're inside the spinal cord. Let's say. All right, this one looks like an excitatory interneuron, and it looks like we've got a direct connection here, right? A synapse. So, what is going to be the result in this neuron would be, a lot, right? A lot of action potentials. Right, maybe it's not quite as many. We'll see why in a second. But it's still a lot of action potentials here. All right, why did we have so many here and we still had a lot but not quite as many over here? Like, why did we lose some? In theory, it should have stayed the same, right? It should have. But, we had a little bit of influence, right? Remember we said in the previous chapter that we could influence the strength of the synapse by perhaps having some other axon come in, right? So this is now we're going to use it. We use that little bit of information that we learned. So let's take an inhibitory interneuron that's capable of creating the IPSP. Like, that's all that neuron does. Good? So if that neuron is told, these red ones, if those red interneurons are told to fire, to release, right, neurotransmitter, always the result is going to be IPSP. That's their job, period. Good? All right, so let's see. We're going to work our way backwards. So if we're generating, let's say, 2 IPSPs onto this axon terminal here. You see how that's gonna make things a little bit less, right? There's our negative influence. So this is why we go from so many to not so many. It's still a lot, right? It's still a lot, but it's not as many. All right, so far so good. Let's see. Where did that come from anyway? Right? So now let's work our way backwards. Oh my, right, you see that? Like this interneuron was told to do that by, that axon traitor, right, over here. And similarly, this interneuron was told to inhibit, right, by this neuron over here. And it just happens that these 2 neurons are on either side, right, of this one that we're pressing on. Good so far? Wow. All right, let's see what else. What else? I suppose that these neurons, right, on either side, they're not, they're not the ones that are getting super pressed on, right? Maybe they're getting a little pressed on because they're next to the point, but they're not getting, like, really super pressed on, right? So they are going to generate some graded potentials. We'll definitely have some kind of right, summation of those. And based on what we know, okay, I guess, all right, I guess they're only going to generate these 4 action potentials. That's all right. That's how much they're receiving. So they'll send their 4, right, each on either side. Let's see. If we go straight, we'll look at the branch next, but if we go straight, all right, we would have our synapse, and oops, looks like we ended up with only 1 coming through. Same situation on this side. Wow, why did we go from 4 to 1? Let's backtrack on that one. Oops, looks like something similar, right, to what we've already discussed, and it is similar to what we've already discussed. Hang on. Let's follow this branch over, right? Follow this branch over to that inhibitory neuron that we have already discussed. So it's this, right? This information that is traveling not only here, but also taking the little side trip to this inhibitory interneuron that's influencing our center neuron. Right? And telling this one, maybe turn something down a little bit, right, in the center. So we've got this influencing this. So far, so good. Now let's look at our final thing, right? Which is this. So we come back to our center neuron. That's the one that we're pressing on really hard. And we've got a lot of action potentials. And when we look at these branches off to the sides, right? Telling these interneurons, right? We should turn that down. Because we've got so much strength here, these IPSPs off to the side are going to be really big, right? Compared to the IPSPs generated here, which are going to be, right? Little. Make sense? So if we've got big IPSPs, right? Going out here, we're really going to turn this down, right? That's why we go from 4 to 1. 4 to 1, right? These IPSPs, not so small. I mean, not so big. Oh, jeez, I can't believe I said that. Not so big, right? Just little ones. So we go, well, we can still have a lot here, right? So now let's compare. We've got a whole lot here. We've got not so many on either side. This is our initial situation. After we get everybody to influence each other, we end up with, we still have a lot in the center. We have almost nothing, right, on the sides. Do you see how we have, in effect, magnified the center by quieting down what's around it, right? All right, so that is that idea of lateral inhibition. We're in effect inhibiting the stuff that is lateral to, right, sort of on the sides of what's in the center. Good? We don't necessarily turn it off all the way, but we turn it way, way, way down. Good? So here's another way of looking at that. Here we've got our patch of skin. We've got our 3 neurons with their receptive fields, a little bit overlapping, right? And this is exactly the same setup that we just saw, just not with the red and the green, right? But you can just think, right? Green, red, green, red, green, green, right? Red, red, green, green. Good? All right, same exact thing. And now let's look at this graph. Aha, the blue line, this one, is what we ended up with. We had a small area right in the center that corresponds to this center neuron that is still above threshold. It's well above threshold, right? But we have, in effect, kind of taken away, right? Taken away the information coming in around that center point. Good? And this purple line is just showing us sort of the original area. If we would not have had lateral inhibition, the output would have been looking more like this. And you see how that, I mean, yeah, there's a big peak in the center, but there's still an awful lot of information, right, coming at you. Because this is all going to the brain, right? There's a lot of information going to the brain, like around the center point. Good? So if we had all this other information coming in around the center point, we would think that, you know, this point, right, would feel more like, I don't know, maybe like this. What good is that, right? For us and our, right, the things that we have to do. This, I don't know, this looks so good, right? We wouldn't be able to do as much stuff. Good? So this setup helps us to do these very fine things, right, by receiving information and being able to really, really localize or pinpoint exactly where things happening. All right, very good. So let's see. Here we just have hopefully a reminder of a little bit of anatomy of our setup. So we've got sensory stuff, coming in, going up to the brain, and we could influence that even as it comes in by sending information down from the brain and maybe inhibiting stuff, right, with that information coming down. So, let's see. We've already seen these two words, divergence and convergence. So now we're seeing that in context with this chapter. And, reminder slide. Remember our map? In anatomy we had a more detailed map, but this is just a reminder. And, this was our anatomy one. Reminder. So just, kind of be sure that you remember. Oh my gosh. Oh! Another reminder slide. All right. So, let's look at some specific receptors in the skin. So this picture, you guys can just memorize the picture. It's pretty straightforward. This is almost an anatomy slide, really. Just a very detailed anatomy slide. So we have a chunk of skin and our different types of receptors. So just, match, right? The names and the receptor type. Some of these we talked about in anatomy. I think we talked about Merkel's and Pacinian, I think, in anatomy, if I remember right. But you can match them up, right, to there. And also, let's add our little bit of physiology information, and we'll add in, what is the type of receptor? Remember we talked at the top about rapidly adapting and slowly adapting. Most of these are mechanoreceptors except for these free endings, right? The ones that don't have these specialized structures around them. And most of the free nerve endings are pain or nociceptors. All right. But otherwise, these are all mechanoreceptors. Some of them rapidly adapting, some of them slowly adapting. So just memorize this slide. And also, most of them are for touch and pressure. We might have a couple of, like here's vibration, right, and stretching. So just kind of work on those. Very good. Very good. Oh. Those are reminder slide of skin. All right. So, let's see. Sort of general notes. General notes about all this somatic sensation stuff. Whatever, just lumping together stuff that we have not quite said yet, to be sure we say all the things. We saw in the picture that we just looked at that a lot of those mechanoreceptors have these, little specialized structures rather than a free neuron. And a lot of those specialized structures are filled with fluid and an old friend from anatomy, collagen, to sort of create that. And when these specialized structures are pressed on, right, that's the whole thing about touch and pressure, is they get pressed on. And that fluid with the collagen changes shape, right? So when you change the shape, that's going to perhaps open the ion channels, right, and the whole rest of the thing that we know. And our graded potential, or we could say receptor potentials. Kinesthesia, this is where your body parts are in space, right? That knowledge and that sensation. We're going to talk more about, like I said, in the muscle control, muscle tissue control chapter. So we've said our little bit about that. We're going to set the details aside for later, for next week. Temperature. So temperature, different temperatures are going to trigger ion channels, and this is a different, different, you could say, a different type of ion channel from what we've talked about before. So far we've talked about ligand-gated, voltage-gated, and mechanically gated. There is another category, which is temperature-gated. I put it in parentheses because, it's its own category. It's a category we haven't talked about yet. We do need to know that, there's this fourth category. But this is kind of the only place we really talk about it, right here. So these temperature-gated ion channels don't play as huge of a role in our course, right? Like physiology course. So that's why I focus on the three main types, ligand, voltage, and mechanically gated, because we talk about those the whole time, right? Temperature gated we only really touch on here. So let's know, okay, they exist, here they are. For the rest of the course, we're going to focus on the other three. All right, good enough, that's fine. Let's see. So we will have some various types of temperature-gated ion channels, and each type is responsive to kind of a narrow range of temperature, right? So you would have one for like ice cold, and then the next one for like kind of cold, right? And then another one for like lukewarm, and so on, right? Hot, all of that. All right. Some of the temperature ion channels can be opened with chemicals, so they would be ligand-gated, especially the hot ones, right? Like the peppers. Good? All right, so we've got those. These particular ones, little to no myelin, so I just kind of embedded a question in there. If we have little to no myelin, fast or slow? Sort of in general. Huh? Little or no? Relatively on the slow side. Relatively slow side, because we've got little or no myelin, compared if we had something with heavy myelin, right? That would be faster. If the diameter were the same, right? Because we also have to, you know, in the bigger picture, consider what is the diameter of each one. But in general, these are going to have little to no myelin, so if you compare these to a different neuron with the same diameter, right, it would be the slower ones. All right, that's all right. And oftentimes, we're going to have pain receptors mixed in with our temperature receptors. And we know that, like if you touch something hot, it also hurts, right? Or if you touch something really, really, really cold, that also hurts, right? So at the extremes, we mix in some pain receptors so that if something hurts, we're going to put that into a reflex arc and pull our hand away, let's say, right? Or pull our foot away because we just stepped on something hot, or we just touched the stove, right? Or touched ice, and that was too cold, right? So that's that. All right. Speaking of pain, here's a little bit of information on pain. We're going to put pain receptors, especially a lot of them, especially in places where there might be some damage, right? That would be serious, and we don't want that to sort of, that stimulation to continue, because there would be too much tissue damage. So let's put a lot of pain receptors there to trigger those reflex arcs to pull body parts away, right? All right. So we already talked about extremes, right? Extremes of temperatures, extremes of mechanical changes, and some chemicals, right, would open up ligand-gated ion channels. Okay, great. Two different types of myelination for two different types of pain. The sharper pains are going to be carried in neurons that have myelinated axons, and then sort of those aching, those kind of pains, right? Unmyelinated, those are the slower ones, but dang, those last long, don't they? Those ones, Alright, let's look at this. You know how if something hurts, a lot of times if you maybe press the area next to it, right? Or rub, right? The area around it, maybe that helps, right? So, that works. That works. Like, that's not your imagination. That's not like an old wives' tale. That's built in because remember all that lateral inhibition stuff and overlapping receptor fields and all this stuff that we've been talking about. Because of all of those setups, we can use that to our advantage when we're dealing with pain, right? And yeah, you can rub on the area that's hurting, and that will, right, help to alleviate some of that pain, right? Inhibit some of that pain. That's good. Good? All right. Let's see. Whoops. Oy. All right. Here's more stuff about pain. Our top situation is just, here's something coming in a pain receptor, and there's our dorsal root ganglion. We enter to the spinal cord and release. That's familiar, right? All right. Alright, don't worry about substance P. We're going to focus on what we've already studied. Right? Glutamate. Old friend. What's glutamate? Let's review. Amino acid. There is no cocaine. Alright. So, amino acid, right? Amino acid. What sort of, we had a subcategory, right? Amino, excitatory. Very good. So you didn't lose anything in an hour. Pretty good. All right. So glutamate. Okay. And then this is just sort of representing going up the spinal cord. And the first place where we have to synapse once we're in the brain, thalamus, all sensory stuff, right? Coming into the brain has to synapse on the thalamus first, right? Except for one location. We'll talk about that later today. And then going up to somatosensory cortex and helping us to say, oh, something hurts in some location, right? All right, so now let's look here. Let's add some stuff. Here we have some neurons coming down. More more old friends. Serotonin, norepinephrine. Woo! What category were those in? Huh? Amines. Maybe there was like one little word before that, just to be thorough. Huh? Biogenic, right? I mean, just to be thorough, right? So biogenic, but that's good. Amines, that's fine. Biogenic amines, great. All right, super awesome. Coming down, telling this particular neuron over here, what is this? Endogenous opiate. We know that, right? To release one of those, right? We had our little list of choices, and maybe that is going to, you see the negative sign, right? Maybe these are going to have these inhibitory effects. Here we're seeing it before the synapse, right, of this neuron coming in. Oh, wow, let's work there. You see that on these receptors, and block the release of the neurotransmitter glutamate, right? This would have been glutamate coming out, right? If we tell this axon terminal, don't release the glutamate. You see how that works? If we don't release the glutamate, we don't, you see they put the X there, we don't get this whole thing. It doesn't even get to somatosensory cortex, so we don't perceive that, right? Sure, there's something happening, but if you block it using our own, right, endogenous opiates like we're seeing here, for some reason, which we can talk about, right, then it's as if nothing's happening in our brain. That's how we perceive it, as if nothing is happening. All right, so let's go back a little bit to our discussion, right, or our introduction last week to things like serotonin and norepinephrine. All right, we talked, let's see, what did we talk about last week that we could kind of overlay onto this? Let's do norepinephrine first. Norepinephrine. What do we know about norepinephrine? Like, where, right? Where, what situations, what system? Let's start with autonomic nervous system. Let's do that. I'll direct you guys. So, autonomic nervous system. Sympathetic, good, sympathetic. What situation are we in? Fight or flight, sympathetic. Boy, especially if we have adrenal medulla kicking in, we're probably in an extreme situation. Maybe there was some big traumatic injury. That would hurt, right? So, to protect ourselves, right? To protect ourselves, we have set up as part of that whole fight or flight situation, especially if it's extreme, and now we've got norepinephrine like crazy, flooding the system. Turn off the pain perception, right? Yeah, the injury's still there. Yeah, you could still be bleeding out. Oh, brother. And technically, that should hurt, right? But we build it into the system that in those extreme situations, we're not going to perceive that, or at least we won't perceive it as much, right? And that is a survival mechanism. All right. So that's where that's coming from. Serotonin we saw last week. That's kind of in a lot of places. That was one of those everywhere, kind of things. All right. But I don't know. Maybe that would play out in different situations. We also talked about, like, when we kind of linked this together, when we talked about endogenous opiates and, let's say, the runner's high, right? Even if you don't run, if you exercise, some other exercise, form of exercise, and that makes you feel good, right? You get to that certain point in your routine, right, or your activity, and you feel good, like emotionally, not the physical part, right? The physical whatever, right? It could be miserable. But maybe you hit a point in your workout where it makes you feel good, right? And so that makes you want to actually do that again, in theory, right? I don't know, maybe not. But in theory, you want to reach that emotional, right, place, that feel good place. So you would maybe exercise again the next day, something like that. So we can use that to our advantage, and that desire to hit that emotional place, right, that emotional feel-good place, can override any sort of pain, right, that you're having, let's say, during your workout, right? Whether it's pain from an injury in the workout or, like, it just hurts me, right, kind of stuff. You can override that and keep going, right? This is why people actually can finish a race on, like a sprained ankle or worse. Finish the race. I mean, so there's other stuff that could factor into that as well, right? Like the determination, and is this like the Olympics or not, or right? This kind of thing. But this plays into it, our perception of, well, how painful is that really? Or can I just run this off? Good. So the body can convince us that it's not so bad. I could run this one off. All right, or I could finish the show, right? All right, I've done that. Oh, really? All right, so there's our setup. And let's have a reminder. I don't know if we talked about this word, but to be sure, analgesia, right, is tamping down on the pain, but still being aware, right, conscious. So that's our official word. Let's see. Oh, brother. All right, what is this? And how are we doing on time, too? Wait, let me see where we are. Hang on. All right, let's just finish this because the next couple slides are review. They're really anatomy review. This is our little reminder about referred pain. We go from this one to this one, right? We remember that from anatomy. Different pain in different organs, right? Our internal visceral organs will be perceived many times as pain in somatic areas. And our classic example is always pain from a heart attack is oftentimes felt not only on the chest, sometimes it's not felt in the chest at all, but in that left shoulder and radiating down the arm. And that situation is referred to as referred pain. And going back now, we see a little bit of a mechanism for that. Here's heart, and we've got a sensory fiber coming from the heart to register, right, if there is pain there, and it's traveling. Remember? See? There it is. I knew I had a picture here somewhere. All right, dorsal root ganglion, and entering into spinal cord, and then going up to the brain. But you see how in this same level, right? Because of course we would have the levels of the spine or spinal cord. So at this level, we also happen to have pain receptors coming in from a particular patch of skin. In this case, happens to be patches of skin, right, starting at the shoulder and radiating down the arm. So because they sort of enter in at this same level, many times there's a little bit of overlap / confusion, right? In the perception, once this gets up to the brain. See how this is like synapsing on the same spot, right? So the brain will perceive that as, even though it's coming from the heart, oh, I think it's this patch of skin, right? I think it's my shoulder. You know, and then people explain it like, yeah, I was working out yesterday, so we did, right? So, all right, so that was this. Let's see. This, I don't know, sort of a reminder, but with additional information, just looking at the actual pathways. Once we've received all this somatic information, we've got to send that up to the brain. We will cross over, right? We will cross over. So when we're talking about neurons that are in charge of pain or temperature, we're going to cross over in the cord. You see the crossover? Right? We'll cross over in the cord and then go up to thalamus and primary somatosensory cortex. When we're talking about our fine touch stuff, pressure, and all that kinesthesia, body positioning, we're going to cross over here in the brainstem. Good? So we cross over later. We're still going to cross over the thalamus and primary somatosensory cortex, which brings up this. This is that homunculus, remember? Homunculus. Do not worry about the details of this picture. That would be crazy, right? We're going to let the neuroanatomy PhD memorize those things. But you guys just know homunculus, and we've got the map, right, on this strip of primary somatosensory cortex. Like, we'll know that much, but we know that from anatomy. Great. So we're going to take our break now, because next is taste. So we'll take our break. Let's go till 10 o'clock. We'll do it even. 10 o'clock. We'll come back, talk about taste and smell. Taste is good. Good. All right.